Okay, let's dive into something pretty fundamental in genetics today. Building maps.
Not roadmaps, though, right?
No, definitely not. Genetic maps. We're talking about figuring out the layout, the order and distance between markers along a chromosome.
And it's kind of amazing how it's done just by looking at how genes get, well, shuffled and passed down through generations.
Yeah, it all comes down to meiosis, that process where chromosomes swap bits.
Exactly. That swapping, recombination, the frequency it happens between 2 points tells us if they're likely stuck together on the same chromosome, linked.
And roughly how far apart they might be.
Uh-huh.
So today we're really getting into the foundational steps. How do you even start building one of these maps?
Right, the very beginning.
And our source material for this is actually pretty cool. We're drawing from lecture transcripts and class notes from Professor Guilherme de Silva Pereira's statistical genetics course.
Oh, nice. That's at the Federal University of Viçosa, Department of Agronomy. Good stuff.
Yeah, so it's like getting a peek inside how the experts actually learn this.
So the mission today then is what? Understanding those first couple of key steps.
Exactly. First, how do geneticists estimate that recombination frequency between markers? That's crucial.
For our value, yeah.
And second, how do they use that R value to group markers? Figure out which ones belong on the same chromosome map, the linkage groups.
Makes sense. Those are the absolute first building blocks.
All right, so let's start with estimating R. Where's the simplest place to begin?
Well, the easiest case to wrap your head around is probably a backcross population.
Okay, remind me. That's when you cross an F1 hybrid back to one of the original parents.
Exactly, one of the homozygous parents.
Okay.
It simplifies the genetics in the offspring quite a bit.
How so?
Because you can basically just look at the offspring and directly count who got the original parental combination of alleles versus who got a new recombinant mix from a crossover event.
Right, like that mouse example in the notes. You see, what was it, 7 recombinants out of 103 mice.
Something like that, yeah.
So you just divide 7 by 103, get your R, 0.068, done. You're literally counting crossovers.
Pretty much. The genotype tells you exactly what kind of gamete it came from. So estimating R is, well, it's just counting.
But just getting the number isn't the whole story, right?
No, not at all. The key question is that R value, say 0.068, significantly different from 0.5.
Why 0.5?
Because 0.5, or 50% recombination, is what you'd expect if the markers were totally independent, on different chromosomes maybe, or just really far apart on the same one. They just assort randomly.
Okay, so we need to know if our estimate is small enough to suggest they are linked, not just random chance. How do we test that? Statistics time.
Yep, statistics time. We need a formal test. The likelihood ratio test, or LRT, is a really common one here.
LRT, okay.
It compares two models. How well does your data fit a model where the markers are linked, using your estimated R, versus how well does it fit a model where they're unlinked, so R equals 0.5.
And you compare the likelihoods.
Exactly. Often we convert that ratio into a LOD score, logarithm of the odds.
LOD score, heard of that?
Yeah, it's just the log base 10 of that likelihood ratio.
Yeah.
And there's a kind of traditional threshold. A LOD score of 3 or more is generally considered strong evidence for linkage.
OD of 3 means?
It means your observed data is 1,000 times more likely if the markers are actually linked than if they're unlinked.
Wow, okay. That's a pretty high bar. Seems convincing.
It is. It's a solid statistical inference.
Okay, so backcross is relatively straightforward. Count, calculate R, test with LRT or LOD. But what about other populations, like an F2, when you self-pollinate an F1?
Ah, yes. That's where things get, well, more interesting, more complicated.
How so?
In an F2 population, some of the genotype classes you see in the offspring are, the term is confounded.
Confounded, meaning?
Meaning, just by looking at the offspring's genotype, you can't always tell for sure if it resulted from a parental gamete or a recombinant gamete from that F1 parent. The information is sort of hidden. Oh. So you can't just count the recombinants directly anymore.
Nope. The simple counting method breaks down completely because you can't directly see every recombination event reflected in the final genotypes.
So, how do you estimate R then, if you can't count?
That's where we need more advanced statistical tools. The notes mention the expectation-maximization algorithm. EM algorithm.
EM algorithm.
Sounds fancy.
It's clever. It's an iterative method.
Yeah.
It basically makes an initial guess about those hidden recombination events. That's the expectation step. Uses that guess to update the estimate of R maximization step and then repeats.
And it just keeps going.
Until the estimate for R settles down, it converges. It's a way to estimate parameters even when some of your data is missing or hidden, like those recombination events.
Okay, that makes sense. A statistical workaround for messy data.
Precisely.
So let's recap the process so far.
Yeah.
You get your marker data, maybe from a backcross, maybe F2.
Yeah.
You estimate R for all your pairs of markers. Simple counting if you can. EM algorithm if it's confounded like an F2. Then you run your statistical tests, LRT or LOD scores, to see if the R estimate is significantly low, suggesting linkage. What's next?
Right. So once you've tested all your pairs and you know which pairs show significant linkage, the next logical step is grouping.
Grouping.
You start putting markers together. If marker A is significantly linked to marker B and marker B is significantly linked to marker C,
Then A, B, and C probably all belong together on the same chromosome.
exactly. Linkage is generally transitive like that. So you build up these linkage groups, which ideally correspond to the actual chromosomes.
But wait, if you're testing potentially thousands, maybe millions of pairs of markers, isn't there a risk of finding linkage just by
That's
chance?
a huge risk. That's the multiple testing problem. It's critical.
Right. If you run enough tests, you're bound to get some false positives.
Yeah.
Things that look significant randomly.
Correct. So you have to adjust your significance threshold. You can't just use that standard LOD of 3 for every single test when you're doing thousands.
How do you adjust?
Well, there are different methods. The notes mention the Bonferroni correction, which is a common, maybe quite conservative one. It essentially makes the required p-value much smaller, or equivalently, demands a higher LOD score to declare significance for any single pair.
To keep the overall chance of any false positive low across all the tests you run.
Exactly. It's a necessary safeguard to build reliable linkage groups and not get fooled by random noise.
Okay, so putting it all together. Estimate recombination are using methods like counting or EM, depending on the population. Test for significant linkage using LRT-LOD, but be really careful to correct for multiple testing.
Yeah.
Then group markers based on those significant linkage signals.
That's the essence of it. Those are the fundamental steps. Estimating the recombination frequency, testing linkage robustly, and then grouping. That's the groundwork.
It's like building the basic street map, right? Before you can get to the fine details of ordering genes or figuring out exact distances.
Precisely. You need this framework first. It's essential for everything that comes next, like, finding QTLs, genes influencing traits, or even helping assemble whole new genome sequences from scratch.
So for you listening, think about this for a second. Isn't it kind of amazing? We look at genetic data from parents and kids, essentially.
Yeah, patterns of inheritance.
And by measuring the echoes of these tiny invisible events, chromosomes swapping pieces during meiosis, we can use statistics to actually deduce the physical layout of genes on chromosomes.
Pretty powerful inference.
It makes you wonder what other hidden biological processes, things we can't directly see, could we potentially start to map or understand if we develop the right kind of clever statistical tools? Something to think about.